All right, so we really just need to talk about, we should be pretty simple. This is not a real difficult chapter, right? We kind of did it within our lab. So we talked about concentration gradients, right? Things naturally move from high concentration to low concentration. We do this because of thermodynamics, laws of entropy, right? In this world, we're always moving towards order or disorder. Disorder. It's how our universe works, right? Organisms, right? We fight against the law of entropy. We maintain high organization, right? We don't allow our molecules to break apart. But in order to do that, we must take in what all the time? Energy to fight against it. So if we were to stop eating, stop taking energy, we would die, and eventually our molecules become more disordered. We would start to decompose, break down, become liquids, gases, and so forth, and diffuse into the world. So the laws of diffusion are really just following laws of thermodynamics and entropy. We're always moving towards disorder. If you watch the quick little clip, right, we have the candle over here, and when you light the candle, those gas molecules are in a high concentration. That's considered a highly ordered state. They're naturally going to bump into each other and spread out evenly around the room, going to disorder, moving around. Okay? All right. So when we do that, we're moving down concentration gradients. We're always moving from high concentrations to low concentrations, and that's passive transport because it does not require what?
Energy.
Energy. Okay. So we already kind of talked about this. The simple diffusion is across our membrane, right? It's simply the small lipid-soluble or non-polar molecules can move through the membrane. Some of them actually can be fairly large, such as steroids, anabolic steroids. They're actually lipid-based molecules, so they can actually move directly across your membrane, compared to some protein-based steroids. Transport proteins, right? We can have simply a protein channel or a carrier protein. Both of them are passive transport, not requiring energy because the molecules are moving from areas of high to areas of low, right? Areas of high, areas low. And in this one here, it's pretty easy to see here. There might be an opening or closing of the gates. Right? It might actually stop or allow molecules to pass. Could there be some energy involved with opening or closing it? Yeah, there could be. But is there any energy needed to actually move the molecules? No, they're moving down the concentration gradient. Just talking about osmosis, just some key terms: osmosis, isotonic, hypertonic, hypotonic. Osmoregulation, just kind of the regulating of water potential in organisms and cells. So you kind of have the notes here, but we can talk about it here, right? So we have an animal cell up top, a red blood cell. We have a plant cell on the bottom. Animal cells prefer what type of state? Isotonic, right? Obviously, we don't want to have our cells shriveled. We don't have a cell wall, so we don't do well when we're expanding. So we really prefer isotonic state, where the concentration of solutes in your cells match the concentration of solutes in your extracellular fluid, in your blood or other bodily fluids. So animal cells prefer isotonic. Plant cells, however, prefer hypotonic, right? Because what type of pressure are they maintaining? Turgor pressure, right? In order to maintain stability, in order to give them the ability to kind of stand up tall and not wilt. So plants prefer hypotonic. Plants prefer hypotonic. Neither cell prefers a hypotonic situation. In fact, both animal and plant cells could die in this situation here. The process that's going on with your plant cell is just a good word to have. It's known as plasmolysis. That cell has been plasmolyzed. And I can show you a quick clip of this at the end. It actually is one of the last pieces of your lab, but it's a lot of setup and it's not really that much to look at. But plasmolysis is simply when the cell membrane starts to pull away from the cell wall. There are connections there, and you can actually start to see, right, the air gaps from the cell membrane pull away from the cell wall. Once they lose that connection, that cell's dead. It's not going to be able to reform those connections. So the plant cell is killed. You can do this pretty easily by putting a plant cell just in salt water, which was the last part of your lab. You were going to take an Eladia cell, look at it under a microscope, put a couple drops of salt water, and so forth. We might be using Eladio later on, so when we do it later on, I might have you just do this as well. But pretty simple. I'll show you a video of it. Everyone okay in terms of, talk about concentrations? I feel like you've already did this in your lab, right? So the hypotonic, the low concentration is outside the cell compared to inside the cell. Water is going to move in. The isotonic is equal concentration, so water is going to kind of be moving in both directions, right? We are never in static equilibrium. We are always in dynamic equilibrium, right? Always in dynamic equilibrium. Water's always moving, but it's just moving at kind of a net zero movement. Here, water's moving in, but does that mean no water's moving out? Water's still moving out. Okay, so it's always dynamic equilibrium. Yep, question? That's when it becomes limp, right? You picture like a wilting plant. That's what happens. It's water regulation. It's no longer in a hypotonic situation. That's why you need to water plants all the time, because they want fresh water around. Because when they have fresh water around, the concentrations inside the plant cells, there's a higher concentration than in the fresh water. Therefore, water is constantly moving in. If you stop watering your plant, the concentration of solids will start to concentrate in whatever water is left. All right, so that's passive transport. If we require energy, we now have active transport, right? Active transport is we are moving against the concentration gradient. We are moving from low concentrations to high concentrations. It requires ATP. Now, the main type of active transport we talk about is what's known as a sodium-potassium pump. You probably had it in normal biology, just a key one to know. But the other thing that I group here, it doesn't necessarily have to be against concentration gradient, but it does involve energy, is endo and exocytosis. It requires energy because it's the moving of the cell membrane to take things in, move things out. But those materials aren't necessarily going against concentration gradients. Okay? But I do group them here. So let's deal with those first. With endo and exocytosis, right? Exocytosis is simply the sending of materials out of the cell. If you remember the question I gave you with the insulin production, dealing with protein synthesis, we produce insulin on the bound ribosomes on the rough ER. They get packaged in the vesicles, shipped through and processed through the endomembrane system, eventually shipped out of the cell. When they're being moved out of the cell, exocytosis, right? Being shipped out via vesicle. If that cell is taking in some type of material, so it's doing like a Pac-Man effect, it's known as endocytosis, taking in. With endocytosis, we really kind of have three main types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Pretty straightforward. Phagocytosis is cell eating. Pinocytosis, cell drinking. Receptor-mediated is just a more specific form of endocytosis, which I'll talk about in a second. So here are just quick little images, right? So here is the cell taking in this larger food particle. It's not necessarily going against the concentration gradient, but it's far too large to go through a transport protein or any other way. So the cell is forming, reaching out with the cell membrane, forming these kind of pseudopods. When the cell kind of forms these arms, they're known as pseudopods. And kind of reaching out around the food particle. The membranes will fuse on the other side, eventually pulling the vesicle in. Easy enough, like the Pac-Man effect. So this is phagocytosis because it's mainly taking in a solid material. Pinocytosis, just a cool picture, right? So here's your amoeba. Here are the pseudopods being formed. And here's the bacteria that's being engulfed by the amoeba. Just kind of cool just seeing how large the bacteria is compared to the amoeba, but the process of endocytosis, or phagocytosis to be more specific. So pinocytosis is cellular drinking. The greens and the purples are not saying solids, they're saying kind of like dissolved materials. So it could be sodium, chlorine, potassium ions. So they're not really solids, they're really dissolved materials, and it's really taking in the fluid. So pinocytosis is taking in the extracellular fluid for the cell. You can see all the little vesicles being formed on the edge, the membrane of this cell. And this is kind of looking at a blood vessel, which is kind of cool. Okay, receptor-mediated endocytosis. It's simply endocytosis that is regulated by a receptor. A receptor is just a protein that binds to specific chemicals. In general, a good kind of biology word that we should have, that we should use, is a word called ligand. A ligand is anything that binds to a receptor. So in our brains, right, we have chemicals that go from one neuron to the next neuron to kind of communicate. What are those chemicals called? Neurotransmitters, right? When they bind to receptors, they could be called a generic term of ligand. Neurotransmitters more specific. So anything that binds to a receptor is a ligand. And when I just put that in, who knows? Okay, so this is kind of looking at receptor-mediated endocytosis. Here, this cell might need to take in some particular type of molecule, and maybe it's taking in a hormone. Maybe this is a muscle cell that needs to increase its growth. Those purple chemicals there could possibly be HGH hormones, right? And the HGH hormones are targeting this particular type of muscle cell. So your body's trying to tell this cell, hey, you need to grow, you need to expand. Right, so that HGH hormone is specific to this cell, and in order to make sure that this cell takes in that particular hormone, we have the receptors to match. So the hormone binds to the receptors, that signals to the cell, hey, we have what we want, we have the molecules that we need, and then it'll undergo endocytosis to take it in. Kind of the interesting, you always kind of see the protein coat. It makes sense to have it because it might be a way for this cell to identify this particular vesicle versus other vesicles that might have been formed. Okay, so receptor-mediated endocytosis is simply endocytosis, but taking in a particular substance. Eric. Absolutely. I mean, they definitely can. Yeah. Not that they have to, but they can. Yeah. Absolutely. I mean, that's a big part of the specificity of your immune system, is being able to identify. Yeah. Okay. So that is active transport because it takes energy, but again, it's not going against the concentration gradient. The real best example of active transport is really the sodium-potassium pump is the most common. And this is my favorite video of all time. Exactly. I'll let you watch it again. It makes me laugh. So they're simply modeling the sodium-potassium pump. Sodium-potassium pump is a protein pump that moves sodium in one direction out of the cell, groups of 3, potassium in the other direction into the cell in groups of 2. ATP is involved, and the cell membrane. So here's our sodium molecules, 3 sodium molecules. Here's your 2 potassium molecules. This is cytoplasm inside the cell. This is the extracellular fluid. We're going to move our sodium out of the cell. This is active transport because we're actually going from a low concentration of sodium to a high concentration of sodium, even though there's no sodium out there, it's high. Potassium is also being moved from a low to a high. So they're both being moved against their concentration gradients. In order to do that, it requires ATP. Here's our ATP molecule. The three people represent the three phosphate groups. If you notice, when they bind to our protein, one of them is going to be dropped off. The protein is going to be phosphorylated. It's a protein. So therefore, when it is energized, phosphorylated, proteins tend to change their shape. In this case, it's going to invert. So it's going to change its shape from being folded this way to being folded in the opposite direction. So as you see, the sodium will bind to the protein. Once that happens, the protein is now available to be phosphorylated. It will be phosphorylated, causing a shape change in the protein, releasing the sodium. Once the sodium is released, it's now available for potassium to bind. Once potassium binds, there's another slight change to the protein shape, which causes the release of the phosphate group. When the phosphate group is released, the protein goes back to its original shape. I'll walk you through diagrams that are a little more clear. I just think this is funny. It makes me laugh every year. So. Sodiums, ATP. I like the guy in the end. He's into it. There go the potassiums. Release the phosphate group, change back to original shape. I also like the group trying to find your place. Wait, hold on. All right. So here's a clearer picture of what's going on with our sodium-potassium pump. We're simply moving our molecules against the concentration gradient. So we have our sodium being moved from inside the cell to out of the cell. So the three sodium molecules bind to our protein. When that happens, that protein, the sodium-potassium pump, is now available to be phosphorylated. So three sodiums bind, it is phosphorylated. Once you phosphorylate energized proteins, they change their shape. So it changes its shape to be inverted or facing the other way. Potassiums are released. It's now available for sodiums are released. Now available for potassiums to bind. Potassiums bind, causing another slight shape change to a protein. When everything binds or is dropped off, the protein is always going to change its shape slightly. So the releasing of the sodiums make this area available to bind potassium. Potassium binds, causing a shape change and causing the release of the phosphate group. When the phosphate group is released, it inverts back to its original shape, putting the sodium into your cell. Against moving against the concentration gradient. Question?
So why is it that sodium is out? So that's just on the food.
Okay. One of the key areas where this is used are in your neurons. And one of the most important things for your neurons to do is to maintain a potential difference. Right? Your neurons maintain an electrical charge. Everyone knows your neurons fire, they have impulses. Well, those impulses are charges. Guess what the charges are? They're sodium and potassium ions. So I know it's weird. What happens is outside, so if this was the membrane of the neuron, the outside will end up being positively charged. The inside will end up being negatively charged. There's some other players there, but if you look at positive and negative, it's differences in charges. So I'm moving 3 positives out for every 2 positives I move in. So outside is getting more positive charges than the inside, even though I'm just moving positives. So this is important in our neurons. When you have electrical impulses, when your neurons are firing, right, and trying to maintain what's known as resting potential, the sodium-potassium pump is what's maintaining the resting potential. There's a couple of little mechanisms in there, which we'll talk about when we get to the nervous system, but this is key there. Yes? What happens to the phosphate group? It's now inorganic phosphate, so it's going to probably head back somewhere near your mitochondria. Once it's in the mitochondria, that is going to, that'll concentrate there. The ADPs will concentrate there, and then your mitochondria will help take the ADP and the phosphate group and make it back into ATP. Right? So the big thing here is we're starting to look at concentration gradients. Diffusion kind of seems like, hey, it's a simple thing. Things move from high to low. Yeah, kind of cool. But in the grand scheme of things, the concentration gradients is what drives all living things. This is the mechanism for exchanging and transferring energy. These potential differences in ATP. So, just to kind of recap, I do have one more thing. Really, we're done. So everything that's going to be on your test, I really just have to have one more slide. I'm going to talk about one concept I'm going to talk about. It's really big picture, but for the most part, we're kind of done for your test. We'll still keep it on Wednesday. Is that good for everybody? Or would you rather have it Monday, so you have to think about it?
Wednesday.
Wednesday? Okay, so we'll keep it Wednesday. Tomorrow? No. A week from tomorrow. But it means that later this week, we are going to kind of move on a little bit. So we'll move on. But what we're going to go into, it might help you a little bit with some potential differences. Things like that. So just to kind of recap, right, we're talking about transport. We have passive transport, does not require energy. We have active transport, requires energy. And that's it. Yay. All right.